Hi everyone, today we're talking about the oxic, suboxic, and anoxic zones of sediment. Specifically in this video, we'll be focusing on marine sediments, but these concepts can be kind of extrapolated and used for other sedimentary environments as well. And first, let me just start by saying that this is part of a series of videos now that I've been doing on my channel over microbial zonation, which is just basically the natural layering or zoning of microbial communities in the environment based on their metabolic pathway. And we talked a lot about that in the microbial mats and biosedimentary structures video, as well as the organic carbon burial and diagenesis video. And in today's video, we're going to be going over all the different metabolic pathways and their significance within the oxic zone of sediments, then the suboxic zone, and then lastly, the anoxic zone of sediments. Let's just jump right in to discussing what these oxic, suboxic, and anoxic zones are and what those terms even mean. Basically, they're pretty well named. Oxic zones or environments are those that contain abundant oxygen. And when I say oxygen, I mean O2, molecular oxygen, like the air we breathe. And here it says dissolved because we're specifically talking about marine environments, and we're talking about oxygen that's dissolved in either the water column or sediment pore waters. And the suboxic zones or environments are those that contain low dissolved oxygen. Now, there are definitions as to the exact oxygen concentration that there has to be in order for it to be defined as suboxic, or maybe you've heard of hypoxic. That's a similar term to suboxic, but sometimes these definitions shift and change and aren't consistent across all references. So you might find different definitions for this in terms of the exact concentration of oxygen, but basically it's low. It's not abundant like in oxic environments. And anoxic environments are pretty straightforward. They basically contain zero dissolved oxygen. So obviously on today's modern Earth environment, there are very, very few environments that can actually be defined as anoxic because absolutely zero oxygen is really hard to come by. But some environments come really close, especially when we get deeper into the sediment of even just regular oxygenated zones of the ocean, because once you get deep enough into the sediment, all the oxygen becomes used up. And so that's what we'll be talking about today, is that microbial activity that uses up the oxygen in the oxic zone, and then everything else they can use to gain energy in the zones below the oxic zone, aka the suboxic and the anoxic zones. And by the end of this video, you guys, you will become very familiar with this bottom left figure here and understand what all of this stuff means and what all those compounds mean and how they're cycled in these sediments. So let's start off with what goes on in the oxic zone. The oxic zone is the uppermost zone of all sediment columns, if it is in fact oxygenated, which most on Earth today are, at least in that upper part of the sediment, right at the SWI, which stands for sediment water interface. The reason it's at the top is because aerobic respiration, so aerobic meaning using oxygen to respire. Later we'll talk about anaerobic metabolism. Those are non-oxygen using metabolisms. So here we're talking about aerobic respiration because we're in the oxic zone, and this is the first metabolic pathway that contributes to organic carbon degradation. So if you haven't seen the organic carbon burial and diagenesis video, I highly recommend watching that one first, or even after this one. I don't think you really have to have seen it for this one, but it's really helpful in understanding where organic carbon is produced, what produces it, how it gets to the sediment water interface, how much of it actually makes it to that interface, and then how it starts to become degraded and undergo diagenesis upon burial. So you can check that video out. I'll link it up here if you want to go see that. But assuming this organic carbon has gotten past all the water column barriers that it faces and has reached the seafloor or the sediment water interface, this aerobic respiration at the sediment water interface is the first thing to tackle the degradation or decomposition of that organic material. And this is for two major reasons. Aerobic respiration yields the highest amount of energy of all metabolic pathways on Earth. And if you've seen my redox stratification video, sorry to plug all my videos here, but if you've seen that one, you know it's because the difference in redox potential, which is something I explained in that video, is the greatest between oxygen, molecular oxygen, and organic carbon. And the greater the difference between the redox potentials of your oxidant and reductant in these redox metabolic pathways, the greater amount of energy that you will get from that metabolic pathway. That's one reason that animals who use aerobic respiration pathway as their metabolism are big and complex and have eukaryotic cells with all these organelles and can do multiple functions and all these things, is because they have more energy available to do that. Other microbes that use other metabolic pathways don't get that much energy from their metabolic pathway, so they can just do the minimum things and be single cellular and not have all these other fancy functions because they don't necessarily need it to reproduce and grow and do their things to be living. The second major reason that aerobic respiration is used first at the top of the sediment and water interface is because they have enzymes that degrade complex organic polymers into simpler ones. And then these simpler ones are what the microbes below them use to break down. And this reason that they have these enzymes kind of also goes back to the fact that they have more energy from their metabolism to have such enzymes and degrade this complex material. So it all goes back to that, and they can do this, and that's the reason that they're first. Okay, moving on. In terms of numbers, around 90% of organic carbon is aerobically respired in pelagic sediments. Now here, pelagic just means basically deep water marine environments, deep water sediments, whereas coastal marine sediments is obviously pretty self-explanatory. It's near the coast. And in these coastal environments, in contrast to pelagic deep water sediments, less than 50% of organic carbon is aerobically respired. That just means that the anaerobes, the anaerobic metabolizing organisms, do more work to degrade organic carbon in these coastal environments. But before we get to these anaerobes, there are 5 main factors you should know that control how deep the oxic zone penetrates into the sediment. The first is the sedimentation rate. The rate at which detrital organic carbon, sometimes also referred to as marine snow, falls through the water column to the seafloor and becomes deposited is really important for controlling the role of aerobic respiration on organic carbon decomposition. And that's something we talk a lot more about in that organic carbon burial and diagenesis video, if you want to check that out. And we talked about how in coastal settings, for example, there is a whole bunch of primary productivity leading to a lot of POC, or particulate organic matter, going to the seafloor at very high rates. And this causes an accumulation or sedimentation rate that's really high, causing the oxic zone to only penetrate to very shallow depths because it can't keep up with that accumulation rate, and all the oxygen is being used up in the water column. And that's also the reason that we see hypoxic zones most often in these coastal marine settings rather than in deep pelagic settings, because these primary productivity or phytoplankton blooms cause the oxygen to deplete so much underneath them that oxygen depletion sets in, and you get hypoxia or suboxia in the sediments and sometimes even the water column that kills animals because they need oxygen. The second major control of how deep the oxic zone penetrates is POC reactivity, or particulate organic matter reactivity. The more reactive the organic carbon, the more easily degradable it is. And if there's a lot more easily degradable material, the oxygen can be used up really quickly, degrading this highly reactive POC and therefore contributing to, again, a shallow depth of oxic zone and a more hypoxic environment because that oxygen is being used up. The third major controlling factor here is the oxygen concentration in the overlying water. Now, this one I'm not even going to explain because it makes total sense and it's self-explanatory. Obviously, the concentration of oxygen in the water column will control also how deep the oxic zone in the sediment can penetrate. It makes total sense. The fourth control on how deep the oxic zone goes is physical properties like porosity and permeability of the sediment. So I talk a lot more about porosity and permeability as well as the difference between the two in that organic carbon burial and diagenesis video, but it's basically pretty simple. The porosity and permeability help control how deep the pore water can go into sediment and how the oxygen dissolved in the water can be transported into that sediment pore water as well. And the last major controlling factor of how deep the oxic zone penetrates in sediment is oxygen consumption by porewater reductants. So oxygen is an oxidant, and we talked about how it oxidizes organic matter, but there are also reductants. And in the oxic zone, the major reductant is typically organic carbon, which reduces oxygen as oxygen oxidizes it in a redox reaction. But other reductants that we're going to talk about here in a few slides that are created below the oxic zone can diffuse upward after they're created or reduced, and then they get to the oxic zone and they can react with oxygen or reduce the oxygen as well and deplete it, because when you reduce something, you're transforming it into something else and it's no longer that thing. Therefore, the oxygen will no longer be oxygen and will no longer be available to oxidize the organic matter, and therefore the oxic zone will penetrate less deep. Finally, moving past the oxic zone to the suboxic zone. In the suboxic zone, we can see in this figure here that there is nitrogen, manganese, and iron cycling going on. Now, the first one we will talk about is nitrogen cycling. That occurs just below the oxic zone typically, and nitrogen cycling occurs via two major processes: nitrification and denitrification. Pretty simple terminology here. The nitrification process refers to when ammonium becomes oxidized to nitrate, and because this ammonium has to become oxidized to transform into nitrate, there has to be an oxidant. And the oxidant typically used by microbes in nitrification metabolisms is oxygen. And so in this reaction, you can see that oxygen is used to oxidize the ammonium into the nitrate, and then oxygen in the process is reduced to water. However, nitrification can also occur by way of using manganese as an oxidant. So this is manganese oxide, a common mineral that can become reduced and dissolved in the process, and that is because the manganese in the manganese oxide is oxidized. It's manganese +4, and it can become reduced to manganese +2, and in this process it oxidizes ammonium to nitrate, causing nitrification. However, the amount of energy gained from each of these different types of metabolisms that are both nitrification but using different oxidants is different. Those using oxygen gain a greater amount of energy than those using manganese oxide, and that just goes back to the difference of redox potential. More energy is produced by the process using oxygen as an oxidant. That's going to be true of most redox or metabolic pathways because oxygen is a freaking strong oxidant. But the second major process controlling nitrogen cycling in this suboxic zone of sediment is denitrification, and this is exactly what it sounds like, the opposite process to nitrification. Nitrate, or NO3- is reduced back to nitrogen 2. So not to ammonium, it actually goes all the way from nitrate back to molecular nitrogen, which is originally what it started as before it became ammonium, and I'll get to that in just one second, so hold your horses. But basically, nitrate can become reduced back to molecular nitrogen by organic carbon, such as glucose as shown here, sulfide as shown in this second reaction here, manganese 2, so just exactly what the product was from the nitrification process using manganese oxide as an oxidant. And there's an opposite process in which manganese 2 can reduce the nitrate back to nitrogen 2 in denitrification. And lastly, iron 2, or ferrous iron, reduced iron, can be used to reduce the nitrate and form molecular nitrogen once again. But there are other processes that complicate the nitrogen cycle, and I didn't really mention them as major processes occurring within the suboxic environment, and that is because the first one, nitrogen fixation, which is that process that I just mentioned that takes molecular nitrogen from the atmosphere and fixes it into bioavailable nitrogen or ammonium or ammonia, that process isn't necessarily occurring in suboxic zones of sediment. By the time nitrogen is in those zones of sediment, nitrogen is already in bioavailable forms, which is why we mentioned nitrification is the first major process in the nitrogen cycle in suboxic zones of sediment. And lastly, for the nitrogen cycle in suboxic sediment, Anomox is a process in which ammonium is converted straight to molecular or atmospheric nitrogen, kind of skipping over denitrification altogether. And although the significance of this process relative to denitrification and nitrification is not fully understood, it is known to occur either by using nitrite or manganese oxide, once again, to convert that ammonia into the nitrogen too. But now jumping into the zone directly below the nitrogen cycling zone in typical sediment profiles, that is the manganese cycling zone. And so this manganese zone is dominated by mostly reducing mechanisms, so manganese reduction in these conditions, suboxic to anoxic conditions, but the products, the reduced products like manganese 2, for example, can diffuse upward into oxic conditions and become reoxidized and then go back down and become reduced, and so on and so on. In fact, it's estimated that for both manganese and iron, each molecule of either manganese or iron is recycled 100 to 300 times before it's finally buried. So how initially is manganese reduced? Well, we just saw an example of how the manganese 4 plus in the manganese O2, manganese oxide minerals can be reduced by way of nitrogen cycling, specifically nitrification. But the most energetically favorable way for microbes to reduce manganese in manganese oxides is to use organic carbon. In this case, the example is acetate. And here I might note that it's important to remember that the types of organic carbon compounds become different as you go down the sediment column. And that is because those easily degradable, highly reactive organic compounds are degraded or oxidized in the first place at the top of that sediment water interface. And as they go down, become less and less reactive and become different organic compounds. And so here you see the use of acetate as organic carbon rather than, for example, glucose, which is more common higher up in the oxic and upper suboxic zone. Getting back to the manganese reduction here, though, we can see that the manganese oxide minerals are reduced and also dissolved during this process. It's something called reductive dissolution. Both manganese and iron oxyhydroxides go through this reductive dissolution process, whereas most other minerals aren't readily dissolved in this burial process like these are, but manganese and iron oxides are reductively dissolved, and we can see here that the manganese 2 produced actually gets diffused upward to the more oxic zone, where it then can be used as a reductant to either reduce oxygen itself or other oxidants in that zone, like, for example, oxidized nitrogen compounds, as we saw in the previous slides. However, some of that reduced nitrogen can diffuse downward and actually form manganese carbonates. But back to manganese oxide reduction, organic carbon and, as we saw previously, nitrogen compounds aren't the only thing that can reduce manganese oxide. Sulfide can also do this, as well as ferrous iron, as well as even iron sulfide compounds. But again, these different reactions, even though they might all produce or yield energy, do so in different amounts. And the most energetically favorable is typically going to be that using the organic carbon as the reductant. Next, getting to iron cycling, which, as we've already seen, is really closely coupled with manganese cycling. Iron hydroxides, just like manganese oxides, like we just talked about, can be reduced using organic carbon. And in this process, iron is reduced from iron 3 or ferric iron to iron 2 or ferrous iron. But the porewater iron 2 or ferrous iron that is produced doesn't always get to diffuse upward into the oxic conditions and be reoxidized. Sometimes it gets used up by the sulfide that's diffusing upward from the sulfate reduction zone that's just below the ferruginous zone. In this zone, sulfate gets reduced to form sulfide, and sulfide can react with ferrous iron to form iron sulfides. The most common of which that you probably know of is fool's gold, otherwise known as pyrite or FeS2. However, iron monosulfide and other sulfides form as precursors to pyrite, and pyrite formation as a whole is really complicated and not fully understood. And I talk way more about the hypotheses behind mechanisms that lead to pyrite formation in my part 3 biomineralization video, as shown here, and I will link it up here in the right corner if you want to go check that out. But now, moving from the suboxic to the anoxic zone in these sediments, let's get into the sulfur cycling zone or sulfate reduction zone, as we just mentioned lies below the iron cycling or ferruginous zone. Sulfur cycling is unique, however, in its relevancy to how much it takes up in the sediment column and how much it controls organic carbon cycling, in that it really varies depending on environment. For example, in coastal marine settings, sulfate is present at 50 times the concentration of all other terminal electron acceptors, or TEAs, combined. And electron acceptors are just things that are oxidants, like oxygen, nitrate, oxidized manganese 4, ferric iron 3, and sulfate, obviously, which is the one that trumps all the others in coastal marine settings. However, in pelagic or freshwater settings, it's very, very limited because there's just not that much sulfate in those settings. But regardless, the bacteria living in this zone, aka sulfate-reducing bacteria, use DSR, or dissimilatory sulfate reduction, to reduce sulfate and produce sulfide by using organic carbon. Another major process that comes along with sulfate reduction and sulfide production specifically is pyridization. Now, pyridization is a really important mechanism for a lot of marine organisms that have to deal with the sulfide that SRB, or sulfate reducing bacteria, produce, because sulfide's toxic, even to those sulfate reducers, because they're like, look, man, this is my waste. I don't need it either. So sulfide being used up to create iron sulfides, like pyrite, for example, is a great detoxification mechanism. And the degree of pyridization that occurs, aka the DOP, is dictated by the amount and reactivity of iron available, or iron minerals available, to react with the sulfide and become reduced and form iron sulfides. And these iron minerals include things like iron hydroxides, goethite, magnetite, and hematite, as well as iron-containing silicates. However, the relative abundance of these different minerals also matters, because iron hydroxides, for example, can easily become reduced, and that iron can become incorporated into iron sulfides because those are very reactive to sulfide, whereas goethite, magnetite, and hematite are much less reactive to sulfide, and they decrease in this reactivity in that order that I listed them. And so their relative abundance matters, because say there's tons of hematite but not much iron hydroxides around, well, you're not going to get that much pyridization compared to if there was a lot of iron hydroxides but not as much of those less reactive iron minerals. You're going to get a ton of pyridization. And the last least reactive iron source for pyridization would be iron-containing silicates. The iron in those is just not really available for iron sulfide formation, especially on the timescales at which it passes through the sulfate reduction zone. However, it might be surprising to you that only about 10% of sulfide produced by bacterial sulfate reduction precipitates as iron sulfides. 90% of that sulfide is used by other microbes that reoxidize it. Yes, that's right, the sulfide cycle is complex. There are tons of sulfur cycle intermediates, and these intermediates between sulfate and sulfide include sulfite, tetrathionate, thiosulfate, elemental sulfur, and polysulfides. And then you can see here some organic sulfur compounds as well, because obviously we incorporate sulfur into a lot of our organic compounds in ourselves, as well as all cells do that, because sulfur is a really bioessential element. And all of these sulfur cycle intermediates go through their own respective reactions that can create energy for microbes in this anoxic to suboxic zone of sediments. What all of these sulfur cycle intermediate reactions, as well as the reactions we talked about in the previous slide with iron and sulfide, show us is that there's a strong interrelationship between the iron and sulfur cycles. And significantly, the burial of organic matter and pyrite heavily control the carbon, sulfur, and oxygen cycles, and have done so throughout Earth's history. Although these coupled processes are only significant in coastal marine settings, which only covers about 9% of the seafloor, these sediments and these processes in coastal marine settings are responsible for about 90% of all organic matter buried annually. And that is a large percentage. And throughout Earth's history, we can see the positive correlation between percentage of carbon and percentage of pyrite or sulfide incorporated into marine sediments. So here we can see a graph showing carbon to sulfur ratios in marine sediments going back all the way to around 600 million years ago, or about the start of the Phanerozoic eon. And what we see is that this ratio correlates with major paleoclimate events and global climate shifts in Earth's history. If you want to know more about this relationship between carbon and sulfur and what we can gain from graphs like this and how we can reconstruct these major paleoclimate events and understand what these chemical signatures mean in regards to these events, I talk a lot more about that and how to interpret these types of data in basically all my oxidation videos, the Great Oxidation Event video, the Neoproterozoic Oxidation Event video, as well as my early Paleozoic oxygenation history video. And finally, we are on to the last cycle in our little sediment column of cycles, and that is methane cycling. This methane cycling zone is typically right below the sulfate zone, and methane cycling is the final step in organic carbon decomposition. Basically, whatever gets past this zone will become buried and ultimately preserved. Methanogenesis, which is the main metabolic pathway of microbes that produce methane in this anoxic zone of sediment, is a pathway in which they use carbon dioxide, inorganic carbon, and reduce it to form methane. And now this is much different than all of the other things we were talking about that use organic carbon as a reductant to reduce other compounds. And instead they're using inorganic carbon and actually reducing it to form a reduced carbon compound that is methane. And this methane that is produced in this pathway can diffuse upward to the sulfate reduction zone, where it can be coupled with sulfate reduction in this process where sulfate reducing bacteria can use the methane as a reductant to reduce sulfate into sulfide. The other major process in this methane cycling zone is methanotrophy. Methanotrophy includes those metabolic pathways that use methane and produce other things. But before we get to methanotrophy, actually, we should probably discuss methane sources. So obviously, yes, methanogenesis is a source of methane for methanotrophs to use. However, hydrocarbons, including methane, can condense to form ice-like crystalline deposits called methane hydrates. And these hydrates represent an enormous sink for methane. In fact, it's estimated that they make up around 2 times of all conventional fossil fuel reserves on the planet. However, this sink could be also a source, because if these things start to melt due to warming of the globe, for example, then the methane release from these hydrates could flood the oceans and atmosphere with this carbon. But methanotrophy, on a more positive note, could contribute to buffering this process as the methane is released from these hydrates. For example, it increases carbonate precipitation, acting as a permanent carbonate sink, and therefore a really helpful process in going forward, as we don't want carbon to be a source to the ocean and atmosphere. We'd rather it sink or become buried and preserved. And so this could be really helpful moving forward, but some can still become oxidized all the way to CO2, which would eventually be released to the ocean as an atmosphere. And although methane is more detrimental on a short time scale than CO2, CO2 is still not great, as we already know. So these are the, ups and downs, processes that could occur, are occurring, and we should all know about. If you want to know more about hydrocarbon preservation, specifically the minerals associated with this preservation and microbes involved and what processes go on to actually preserve, ultimately, in the rock record, hydrocarbons, you can watch that video, which is coming soon. And then I also have the prebiotic Earth and earliest life videos. I'm currently editing those because I'm filming these out of order, and I think it's going to be 2 videos because it ran really long as well, probably just like this one. Anyway, I don't want to ramble on. As always, the major reference I'm using to make this video is the Introduction to Geomicrobiology book by Kurt Konhauser. This is a really great book. It's linked in my description if you want to check it out, as well as other minor supporting references are down there too. So with that, thanks so much for watching, and if you want to suggest any topics for future videos down below, you can do so in the comments. And I will see you guys next time. Bye!